Proteins are simply polymers that are assembled from amino acid units as the monomer. They can be a homopolymer, one amino acid, or as they are found in native form, they can be multiple amino acids, all sorts of different types, linked together in different sequences. And those sequences are what determine function. Those 20 natural amino acids that I show you can make up literally thousands and thousands, maybe millions of proteins. And there's a problem that you will see that addresses how much variation you can get. These are linked by the peptide bond between the carbonyl of one amino acid and the amino group of the next amino acid. And these peptide bonds are what hold proteins together. They form the primary structure. And you can see that the backbone is simply a repeating NCC, NCC, amino, alpha carbon, carboxyl, amino group, alpha carbon, carboxyl, and so on for the length of the protein. But they have these side chains, and each amino acid brings a unique side chain, and that uniqueness allows special and variable properties of the protein, and specifically the ability to fold into three-dimensional structures. Proteins are written from the N-terminus to the C-terminus, and the amino acid sequence is called the primary structure of the protein. It is the primary piece. What is the order in which these amino acids are linked? And that N to C terminus way of writing and thinking about proteins derives from the fact that they're actually synthesized amino to carboxyl. And they are always written in this orientation. They're synthesized in this orientation, and they're written in this orientation. And protein sequence is often written as a sequence of 1 or 3-letter abbreviations. So these are abbreviations for glycine, lysine, proline, glutamate, serine, tryptophan, glutamate, glycine. And the 1-letter abbreviations and the 3-letter abbreviations. And that simply reflects these R1, R2, R3, R4. Glycine is R1, lysine is R2, proline is R3, glutamate is R4 in this sequence. And this is the way in which you will see protein structure presented. But it turns out you have to be able to do something. If it were just a linear polymer, it's just like DNA. And so it was known that proteins formed structures and that they were varied. And in the 1930s, William Asbury took wool and hair, which are protein, and using X-ray fiber diffraction, he showed that there was a coiled molecular structure that he called alpha. This later became the alpha helix. When he heated or stretched the fibers, he found another pattern of diffraction that he designated as beta, which later became beta structure or beta sheet. And so here is the alpha helical structure that was identified by Pauling. You have a historic article, and there's a YouTube that will be available in your resources. And you can see both a side view and a down the helix view of this structure. Notice that this structure is stabled by hydrogen bonding, and the hydrogen bonds are circled and highlighted here. And those hydrogen bonds are between residues that are separated by 4 amino acids. So if this is 0 is the amino group of the amino acid that has the 0th carboxyl, then the next amino group is 1, the next one after that is 2, the next one after that is 3, and the 4th one is hydrogen bonding to the carbonyl of the 0th amino acid. So backbone hydrogen bonding between residues that are separated by 4 residues. And notice that this hydrogen bonding, this alpha helical structure, is within the same strand. It is a close interaction within a single region of polypeptide strand. You can have interactions of the R groups because this is a polyalanine sequence. You'll see that there isn't a side chain really to interact, but with some of the longer side chains, you can have interactions of the actual R groups, the side chains, that also stabilize the helix. The other type of secondary structure is beta sheet. In this case, the backbone hydrogen bonding is between the strands. So you see that the hydrogen bonding is between strands, and these strands can run off and do other things. They can be entirely separate strands and form a beta sheet structure. And again, there's a YouTube on beta structure. Now, note that there is not only the interaction between the amino group and the carboxyl group, but also between the side chains, between the R groups. And you might think for a minute about, does this limit the size of the R group? And also, could proline participate in this structure? Now, it turns out that the orientation in beta sheets can be either anti-parallel, shown on the left, or it can be parallel. And you might notice that there are some differences. And you might think just for a minute about whether those differences would result in distinctions in the stability, knowing that hydrogen bonds that are linear are stronger than hydrogen bonds that are kind of bent. So in fact, there is higher stability and the possibility of R group interaction in the anti-parallel. The R groups are aligned so that they can interact, and the NH and oxygen on the opposite strands are aligned to form a more stable structure. Nonetheless, there are definitely examples of parallel. Notice that both strands go NC, C, NCC, or NRO if you're just looking at the letters on both sides. And note that the hydrogen bonds are at an angle, and there is not the opportunity for the R groups to interact. And so there is lower stability, both due to the geometry of the hydrogen bond and the absence of R group interactions. And you can see how easily the anti-parallel forms literally a sheet, which is the reason that these are often called beta sheet structures, because you will see a sequence of these. Sometimes it's just two, but often 3 or 4 or more strands will form a structure that can have very significant stabilities, and these can stack on one on top of the other to form sort of a stacked sheet structure. And again, the hydrogen bonds are linear, and there are 2 sets of them that form, and then you have the R group interactions on top of that. And if we sort of blow up the beta sheet structure, this is a 3-dimensional structure from crystallography, and you can see the red oxygen and the hydrogen bond outlined in yellow. Some of the hydrogen bonds in the NHO orientation are obscured, but you can see those, and I've highlighted them also in the structure on the right. Here are the R group interactions, and so you can see that the R chains on the outside and a few of the R group interactions on the inside of the structure. And you can see that it's pretty crowded on the inside, so you're actually limited in the R groups that can be accommodated in a beta sheet. And if you have an extended beta sheet, you definitely have limitations, because the R groups on both sides of the strands have to be able to be accommodated within the small space. And you'll see that this is an anti-parallel structure. There are actually gray arrows underneath, if you can see them here, that show the orientation of these, and you would know also just by the orientation, the linear orientation of the hydrogen bonds that are highlighted in green, that this is an anti-parallel structure. So why do you think that alpha helix and beta sheet are each referenced as secondary structure? So we've talked about the primary structure is the sequence of the amino acids. Why is the secondary structure? And how does the energy of these structures compare to the energy of the peptide bond? These are held together by primarily hydrogen bonding interactions between the NH and the CO, with a little contribution from the R chain interactions. So how does that compare to the energy of the peptide bond? And what does that mean about stability? When you think about these structures, what contribution does the side chain make to the secondary structure? Is it key to the stability of these secondary structures? And would you expect all amino acids to participate in secondary structure? Will all amino acid sequences be able to form alpha helices and beta sheets? Again, I urge you to look at the videos on the alpha helix and beta sheet because they can rotate them in 3 dimensions, and I think it enhances your ability to see and understand and think about these structures, which are very important really for the rest of this course and into all of biological sciences.